Hey everybody, we are going to continue on with From Gene to Protein, and we are going to start talking about translation and what materials are necessary for translation to occur. So translation is the way that a cell interprets a genetic message and then builds a polypeptide from that genetic message. So you're going to need a few things in order for this to happen. First, you need tRNA or transfer RNA, you need ribosomes, and you need mRNA, so that messenger RNA that was made during transcription. So transfer RNA is the same thing as when you see tRNA. It's made by transcription. It's about 80 nucleotides long, and this is what carries the amino acids that are used for that polypeptide synthesis. So the tRNA transfers the amino acids in order for translation to occur. So what does tRNA look like? It has actually some areas that are double-stranded. So I know before we said that RNA is single-stranded. tRNA sort of loops around, so it actually has some areas that are double-stranded, and then it's got 3 loops. And then the amino acid attachment site is at the 3' end. And one of these loops is going to serve as the anticodon. So it's going to be 3 bases that correspond to the mRNA codon. So this, I think, does a pretty good job of sort of showing you what this all looks like. So here is a 2D version of tRNA. Here's your 3' end, and so this is going to be the amino acid attachment site. Now, if you go from 3' to 5', you can see that there's a region right here where, because it's kind of looped around, you have corresponding base pairs. All right, and then you've got your first loop, you've got, again, corresponding base pairs, and then you've got a second loop. This is going to be your anticodon, so this is the part that matches up with the mRNA. Again, because of that loop, you've got some corresponding base pairs here, and then going around into your third loop, and again, because you have these areas that are looped, you end up, even though it is one strand that's folded like kind of in on itself, there is some base pairing because there are regions that appear to be double-stranded. A lot of times you're just going to see the symbol written like this. You see that a lot in your book and in this lecture. So the anticodon is the region of tRNA that base pairs to the mRNA codon, and it's going to be a complement to the mRNA bases. So it's usually going to read the same as the DNA codon. I say usually because there's going to be a couple differences. The big difference is that anywhere where you see in DNA a thymine, in the tRNA you're going to see a uracil. So if you don't have a need for a T in the DNA, so here's your example, the DNA triplet or codon would be GAC, the corresponding mRNA codon would be CUG, and then the tRNA anticodon would then be GAC. So it is actually the same as that DNA triplet. We've talked a little bit about this wobble effect and about how that third base pair in the codon, there's a little bit of room for error. And so the wobble effect allows for 45 types of tRNA instead of 61. And the reason for this is that in the third position, you can pair actually with A or G. We've also looked at the amino acid chart, and you've seen that a lot of times you have overlap, and so, you know, if sometimes that last base pair, it can be A, U, C, or G, and it's all going to code for the same amino acid. You also have inosine, which is a modified base in the third position, and when that's present, that can pair with U, C, or A. So we do have some modifications that kind of play into this wobble effect that we've talked about, so just understand that in that third position, you do see a wobble, you have a little bit more room for error, so U can pair with A or G. You have inosine, which can pair with U, C, or A, and again, that is a modified base pair that can be in that third position. And the importance of this is that it allows for fewer types of tRNA, and it also allows for some mistakes to code for the same amino acid. So you could have some mistakes, but then get the same polypeptide even though those mistakes are present. Okay, so next we have amino acyl tRNA synthetases. These are going to be a family of enzymes that add the amino acids to those tRNA molecules. The active site is going to fit one amino acid and then one type of tRNA. So it uses like a secondary genetic code to load the correct amino acid to each tRNA. So this is what you're looking at here. You can just see that you've got the amino acid tRNA synthetase the enzyme. You've got room for the amino acid to fit in there. You're dropping off some energy. Once that happens, the tRNA will come in. You'll lose an AMP, and then you'll have your amino acid tRNA, or your charged tRNA that carries that amino acid on it. Next, we need ribosomes in order to go through translation. And so ribosomes have two subunits. They're made in the nucleolus. It's about 60% rRNA and about 40% protein. So rRNA is ribosomal RNA, and this is going to be the most abundant type of RNA that's found in a cell. Here you're looking at a picture or a diagram of the large subunit of a ribosome, and so you can see you've got proteins and you've got RNA. Looks kind of like a big mess all jumbled up together. And here you're looking at both subunits. So you're looking at the large and the small subunit together. So the large subunit has 3 sites for tRNA. The first site we call the P site is the peptidyl tRNA site, and this is going to be the what carries the growing polypeptide chain. Then you've got the A site, which is the amino acid tRNA site, and this is going to hold the tRNA carrying the next amino acid to be added. And then E is the exit site. And so this is going to make more sense right now. So here is your computer model of a functioning ribosome. Here you have something that's a little bit easier to understand. So the top part is going to be your large subunit, the bottom part is your small subunit. You've got your E site, which again is the exit site, your P site, and then your A site. And so what happens is the mRNA is going to run through this ribosome. So you can see the 5' end has already been read, and now the 3' end is sort of still flowing through that ribosome. And so you have 3 base pairs at a time that are going to be at each of the sites, the E, the P, and the A. So those are your codons. You can see here in the P site that this tRNA has brought in its amino acid, but it's also holding the growing polypeptide, or that growing amino acid chain. And now in the A site, you have a new tRNA come in that has a new amino acid that is not bound to anything. And so once these two are next to each other, you will have a peptide bond form in between these 2 amino acids. Once that happens, you will see this tRNA released because its next site is the exit site, so that's where it leaves. And this will move over to the P site because it will be carrying that growing polypeptide chain. So I think the easiest way to remember it is the E site is the exit site. That part makes sense. P site holds the polypeptides. A site is the new amino acid site. All right, and as the mRNA is moved through that ribosome, these will flow from one site to the next. So your lecture question for today, can you guys explain how ribosome structure is important to translation? So send me a quick message with your answer to that question. Thanks, guys.